from Australia, giving our Terasaki lecture series. He received his bachelor's and master's in chemistry from University of Washington in Seattle, 99 and 2003, PhD in chemistry from University of New South Wales in 2007. He was an NIH postdoctoral fellow at University of Chicago. Then he was an assistant professor and associate professor in Urbana-Champaign till 2018. Then he joined the UNSW Sydney in 2018. He received many awards, including the Cornforth Medal from the Royal Australian Chemical Institute, NIH Ruth Kirstein National Service Award. He was a Cowley Fellow in 2014, NSF Career Award, Young Innovator Award, and many other awards. His research interests include design and development of extracellular matrices and dynamic hydrogels for cell engineering. The floor is yours, Chris.
All right, great. Well, thank you so much, Mehmet, and thank you, Ali, for giving me the opportunity to present in these wonderful seminars. I wish I could be there in person, as I said, but alas, we have to do these things remotely, being on the other side of the world and all. And so today, what I'd like to do is tell you a story about really the bioengineering type work that we've done over the years, from a chemist perspective. And so I'm a materials chemist at heart, and so that was my training. But I've moved more into the world of biomedical engineering and designing tissue models and trying to find new and innovative ways to tune extracellular matrices in order to get cells to do things that we want them to do towards translational outcomes in the end. And so essentially, this is where I reside. And so, as Mehmet mentioned, I was at University of Illinois for a number of years. But while I loved the small town vibe, the beach was calling. So I moved to UNSW Sydney. This is Australia. We're very far away. So for me, it's about 7 a.m. To I believe it's 2 p.m. in Pacific Standard Time. And these are some shots of UNSW. It's really a wonderful place, a very large campus. We're part of the group of 8, which are the 8 largest student-serving universities in Australia. And one of the nice things about UNSW Sydney is we're really close to the beach. And so this is Coogee Beach. It's really only a 20-minute walk from campus. And so, if you come and study at UNSW, you'll have opportunities to read your journal articles on the beautiful sand. That's what I did. I studied there many years ago. Okay, so I'm going to launch right into it, and I'll start with a provocative question that I often ask myself, and I think that we as bioengineers and tissue engineers ask ourselves. And so, what are the things that we need to make a tissue? And related to that, central question is, how does nature do it, and can we learn from nature? And here we have an example from some of the just really terrific work from Jennifer Lewis at Harvard University. I'm always very inspired by Jennifer's work, but of course there's many others, including Ali Kadam Hosseini and Adam Feinberg and others who will talk a little bit about their work today. Really the way that we've envisioned these types of things is utilizing some advanced biofabrication techniques where if you engineer a bioink and you have the right types of cells, then bringing these things together, we should be able to fabricate tissue mimetic architectures and establish form and function of these things. And we can use various techniques to print inks and make channels. But I'm going to suggest that nature does this a lot better. And one of the reasons I think that nature has advantages currently is that over millions of years, evolution has led to this really intimate crosstalk between the materials and the cells. And this is the thing that gets me really excited. And the reason as a materials chemist, I have become fascinated with biological systems. Because if you look at just some of the beautiful hierarchical structures that occur in tissue, this is all driven by a crosstalk and a feedback between materials properties, and so that can be the chemistry and the viscoelasticity of an extracellular matrix, and cellular activity, the way that cells feel that material and respond to it through various means, and subsequently remodel their extracellular matrices and deposit materials to make functional tissue. And so, can we utilize nature's materials to, essentially build design criteria for our biofabrication approaches? And one thing that I'd like to highlight is that oftentimes when we do this, we tend to not make the cells front and center. And what I mean by that is, we can isolate the right cell type. We usually grow them on tissue culture plastic because this is the infrastructure that we've been given. And these cells often lose their identity. And we talk about that with respect to their propensity to differentiate, their ability to remain multipotent if it's a stem cell. But it's more than that. Cells adopt a very complex heterogeneity during the developmental process. And in so doing, they respond in ways that are tightly orchestrated by the microenvironment. And so we need to think about the cells. And so, what do we need? Well, scaffolding. A lot of my colleagues would say that scaffolds aren't essential. If you give the cells the right microenvironment, they will synthesize their own matrix, but we believe that scaffolds serve a purpose at certain stages. Scaffolds should be bioactive and have a well-defined architecture that can be resorbed. And with respect to the cells, we need to consider that it's the appropriate source and type. And so this could be whether it's patient-derived autologous cell or whether it's some biobanked cell type. But then something that I've really grown to think quite deeply about is heterogeneity and having a desirable heterogeneity. Oftentimes we're thinking about combating heterogeneity, but I would hold that understanding why cells are heterogeneous in a population and what these characteristics that are adopted over time lead to with respect to functional activity. And so we want to get these cells to be like the cells that are in a native tissue. And often this is quite a challenge. So this is how we do it as biomaterials scientists. The concept that I think drives a lot of our work is if we have some in vivo cell state that we want to recapitulate through some means, rather than growing it on tissue culture plastic, biomaterials engineers have developed a whole suite of methodologies to design an interface to get the cells to be more like their native counterparts. And so this involves nanopatterning, micropatterning, using viscoelastic matrices like hydrogels where you can control the stiffness robustly. And in all of these platforms, what we're trying to do is give the cells the cues it needs in order to remain a certain cell type or get it to differentiate towards some interesting lineage. And so what we do in my group is we keep this front and center in our efforts, and then we work to design material systems that can control this in vitro. And so I'd say the majority of our efforts are really on studying cell biology using advanced materials. And so one aspect of that is dynamic extracellular matrices. And so in this area, we do things like change the dynamic properties of hydrogels in 2D as well as in 3D so that we can understand how cells engage with the matrix. Another work that we've done quite a bit on is in stem cell engineering. And so if we have these nice defined matrices, we can tune the microenvironment to trigger epigenetic changes in the cell to get them to undergo various transformations that are useful to us. And this is both of these projects are work that I did at University of Illinois, Urbana-Champaign, and really led by Junmin Lee, who I don't know if he's in the audience, but currently at the Terasaki Institute doing a postdoc. And June really did some outstanding work in getting these things off the ground. So another work that June did was involved in mechanochemistry, and we've now taken this to new levels where we've found methodologies in which we can tune linkages within hydrogels to be biomimetic. And so the idea here is nature has evolved all these complex three-dimensional biopolymeric architectures that can respond in well-defined ways to things like applied force. Can we simplify these using synthetic chemistry? And so the idea, as an example here, using a disulfide link gel where this sulfur-sulfur bond is significantly weaker than a carbon-carbon bond, such that force may be able to lead to breaking and healing of that bond. And can we associate dynamic activities with that? And then finally, an area in the group that, that we've been focusing quite a bit on recently is in additive bioassembly. And this is biofabrication. Now, I presented right at the start this idea of having to think about the cells and the materials. And this is where the term additive bioassembly has become quite attractive to me, because we're not just assembling materials and chucking cells in there. This is, I think, a lot of the early work went, but we're actually designing a material that can trigger cells to have very important activities. And so it's an assembly process, and it's a stepwise process where the materials tell the cells what to do, and then the cells then remodel the material or restructure the material to further augment that activity. I hope that's clear. It's somewhat of a convoluted picture, but that's how nature does things. And at the heart of all of our work is cell plasticity. And so probably a decade ago, I started getting really interested in, this black box of mechanotransduction to the nucleus. And at the end of the day, it's absolutely true that the materials in which a cell resides dictates the downstream signaling that ultimately regulates the epigenome and gene expression. And so that really is a central driver in our work. And so today I'm going to talk a little bit about both sides of the coin here. I'm going to start with telling you a bit about some stem cell engineering, and then I'll show how we've applied these ideas of cell engineering to biofabrication. And when we talk about cells and how we can engineer these cells, many of you are probably familiar with Waddington's epigenetic landscape. And as a chemist, we often think of the reaction coordinate and activation energies that you need to surmount in order to make a chemical reaction proceed. And there's some very interesting analogies in developmental biology. And I think this idea of the energy landscape in the epigenetic state has been quite intriguing. And so here's the way the picture has been formed. The idea is that here at this valley, you have a pluripotent cell that can form all of the tissues in the body. It then differentiates into a more committed cell type. And so this is where the adult stem cells reside. And then further slides down this landscape into a valley that is a somatic cell state. And the question that we're often interested in is what promotes this process of differentiation? But in addition to that, what then promotes things like transdifferentiation? We now know that in many contexts in biology, cells can go from one somatic state to another, and even dedifferentiate, where a cell that we've formerly believed to be committed for all times to a particular type of cell, in the right context, receiving the right microenvironmental stimuli, these cells may be able to dedifferentiate. And the classic example of this is in earlier life forms like salamanders and axolotls, where if you cut off a limb, they're able to dedifferentiate cells at that wound site and regrow that limb. And so wouldn't that be a wonderful paradigm for regenerative engineering? And so, when we talk about clinical applications, these are the main types that we often focus on. So pluripotent stem cells, they can come from many different sources and have the propensity to undergo all of the lineage programs of an organism. And then often we start earlier or downstream rather, and look at adult stem cells. These could be neural stem cells, or the more commonly utilized mesenchymal stem cells that could be isolated from many different tissues. And we've worked quite a bit in this area with MSCs. And so I'll tell you a little bit about some of the stuff we've done here. And I'm pretty sure everyone in this audience is very familiar with these types of cells. But just to give a quick primer of their importance, these mesenchymal stem stromal cells have been derived from multiple tissues, bone marrow, adipose tissue, etc. And they have the ability to differentiate to mesenchymal lineages like chondrocytes, osteoblasts, adipocytes, with even some evidence of transdifferentiation to
we can't hear you, Chris.
I'm sorry, everybody. It seems that I got kicked off my internet. Let me see if I can get back into it. I hope we're okay.
Yep, it's good.
Okay. Yeah, it's got a bit of a dodgy internet here in Australia. So I'll continue. And so, there's lots of potential for these cells to be utilized for therapeutic intervention. However, I think one thing that's been a bit of a struggle is that these cells have run into a lot of difficulties with respect to clinical translation. And I think a lot of that is because these cells come from a sorry about this. I'm trying to get it to change slides, and there we go. There's been broad variable in efficacy. And so one of the focuses of my group has been, well, if you're injecting a autologous or allogenic cell type into something like an infarcted heart, where you have a very different microenvironment, often very hypoxic, it's not surprising that a lot of these cells will die. In addition, when you're culturing them on plastic, you can really influence their propensity for different lineage specifications. So one question that we ask is, well, can we optimize cultures to essentially prime or normalize activities in these cells? And I've been collaborating over the last couple of years with this company, Sonata, down here in Melbourne, Australia, that's developed this Chimeris platform, which is a technology where induced pluripotent stem cells have been differentiated to form a mesenchymal stromal type cell. And these cells have several advantages, because if you have just one source of cells that are allogenic and can be biobanked, then this is in many ways beneficial for therapeutic intervention. And so we started exploring how materials can influence mesenchymal stromal cell secretion some years ago. And this is work by Amr Abdeen with some help from Junmin Lee. And what Amr and Jun did was they utilized a protein conjugation of hydrogels such that you could vary the stiffness of the material and then study how that influenced the cell morphology and its characteristics, but also how it influenced its secretion. And this paper that now is 7 years old, how does time fly? We demonstrated that on precise matrix conjugated materials, we could trigger a high degree of secretion of molecules that were immunomodulatory and proangiogenic. And this was shown in a secondary assay of tubulogenesis, where we have microvascular endothelial cells on microgel. And when we added conditioned media from mesenchymal stromal cells cultured on this 40 kilopascal fibronectin, we saw enhanced secretion. And so this is how that assay looks. And so you take cells, MSC media, and this is an old animation that Amr made me some years ago. It's very fancy, I know. And you take that conditioned media and you put it on the endothelial cells, and you see how they form tubes. And then using some pretty straightforward analysis programs, using ImageJ and others, you can quantitate the degree of tube area. And so we've been doing this with these Chimeris cells from Sonata in Melbourne. And what we found is that very particular types of matrices can promote enhanced secretion of proangiogenic molecules as measured by the degree of tubulogenesis. And so in particular, some of the collagen conjugated hydrogels, and these are polyacrylamide hydrogels, and the fibronectin conjugated materials. But one of the things, excuse me, that we've been really intrigued with is, since we can really prime and activate these cells to be to have a therapeutically interesting secretome, something that has been bugging me for a few years is, well, is this something that we need to do every time we prepare these cells for in the clinic, or is it something that can be cryostable? And so we've been exploring how we may be able to preserve these phenotypes. And this is an example of some of our tubulogenesis results, where you see that the negative control, where you don't have any growth factors, you get very low tubulogenesis. Positive control, where you're adding VEGF and PDGF and all of these growth factors, you get high. And then you can see how the conditioned media influences this. And so we then took these cells that had been secreting these potentially efficacious molecules, and then we froze them down for a month. And then we took the cells out of cryo and just cultured them for 12 hours, and then assessed their secretome again. And remarkably, many of these hotspot conditions, the 1 kilopascal collagen and the 10 kilopascal fibronectin in particular, showed enhanced maintenance of the secretory phenotype that we thought was useful. And we've gone on and profiled this. And so using a cytokine array, we're able to look at a range of growth factors, and we see upregulation of many, many different immunomodulatory and angiogenesis promoting growth factors from these cultures. I think it's just absolutely stunning that we can cryopreserve this. And this is important because it suggests that for manufacturing cells and for production of therapeutic stem cells, that this is a viable approach to culture them on these designer hydrogels and then freeze them down. And so this might be a new pipeline for preparing cells. But one of the things we're curious about is, will this be maintained for in vivo experiments? And so we did this chick chorioanatomic assay, where you take the membrane of a developing chicken embryo, and what you see is a neovascularization after you apply the cells to that membrane, and this is quantitated down below. And just remarkably, when we take these cells out of cryo, we see nearly identical results, where we see the same degree of angiogenesis within the chick embryo when we take these cells from cryo. So I think this is a really potentially exciting approach to normalize and baseline these things. And with respect to a lot of the concerns with cell death, I also believe that we can optimize culture conditions to hardwire these cells to be more accommodating of these types of microenvironments. And so I think in the field of stem cell engineering, this is an exciting new approach. Okay, and so that's what I wanted to show you today in the stem cell engineering work. I'm going to shift gears a little bit and tell you about some of the work we've been doing with iPSCs, because I think that these have really taken center stage as a model cell type for developing tissue analogs for drug development in fundamental biology, as well as being a source of cells where we may be able to differentiate to every tissue in the organism. And so, these cells have a lot of promise for regenerative therapy, being that they can differentiate it to all of the different cell types in the body. But one of the things that I've been interested in for a number of years is some of the early events that are triggered in the development of the embryo and the so-called models of embryogenesis. And so one of the earliest stages after the blastocyst stage is this process of gastrulation, where the early embryo starts to specify the 3 germ layers in well-defined arrangements. And at the start of this process, there's the evidence of this primitive streak population that's positive for T brachyury and SOX17 for mesoderm and endoderm, as well as markers for SOX2. And there's a very interesting organization. And I've always been interested in form as it underlies function. And so one of the things we've been exploring recently is, can we use these platforms where we tune the biomechanics and the biochemistry at that interface to direct some of these differentiation outcomes in cells, ultimately for clinical applications, but also for fundamental interest. And so, using some of our micropatterning approach, Pallavi Srivastava, a student in the group who's just done an amazing job at pioneering some of this developmental work in our lab, she asked some very simple questions. When you look at the majority of literature, when people have explored these pluripotent stem cells, these things are cultured on very rigid glass substrates or plastic. And even when utilizing things like microengineering, oftentimes these material systems are, again, used on very rigid materials. And so what Pallavi wanted to explore is, how can things like geometric confinement using micropatterning on hydrogel materials, how could this influence these cells' ability to initiate some of these differentiation events? And so she started micropatterning these cells, and you can see here on the top, this are cells on glass. And we have just our free cells and colonies, and as well as ones that have been micropatterned on glass using standard approaches. And you can see these cells have very robust staining for OCT4, which is a pluripotency marker that's commonly used to assess pluripotency. But then one thing that was quite interesting is that when we did this micropatterning on our hydrogels using our hydrazine protein conjugation chemistry, what Pallavi noted was that, particularly in regions where you microconfined these populations of pluripotent cells, we saw loss of OCT4 in the center, where you saw this uniform ring of the pluripotency marker OCT4 at the periphery. And so this begged the question, are these cells undergoing some type of differentiation? And if you you stain them for things like SOC17 for endoderm, as well as some of these other differentiation markers, what we see is that the cells lose OCT4 and at the same time start to gain expression of endoderm markers for SOC17. And so this is quite interesting because it really suggests that something about confinement on these more biophysical microenvironments that could be more relevant to physiological systems, you can trigger these types of initiation events and differentiation. This isn't altogether surprising considering some of the really seminal work that's been done over the years demonstrating how the viscoelastic properties of the microenvironment can lead cells to differentiate. But no one had shown that materials can trigger this on their own. And so we started exploring that a little more thoroughly. And if you do gene expression analysis of these microconfined cells, we see that on glass, we have upregulation of all of these pluripotency markers. When you pattern these cells, you see that they maintain a lot of the pluripotency when they're on glass, but they start to turn on genes associated with various germ layer markers. And some of the work by Matthias Lutolf and Jianping Fu and Warmflash have shown quite elegantly that confining these cells on glass can lead to some very precise patterning effects that influence their differentiation. But the thing that we found was quite striking is that when you culture them on hydrogels, you lose that pluripotency out of the gates. And you start to see increase of expression of markers of differentiation, as well as things like epithelial to mesenchymal transition, a hallmark of gastrulation, and various mechanotransduction pathways. And so we've done quite a bit of work to try to characterize this. And at the end of the day, we found that the wingless type, so-called Wnt signaling, seemed to be front and center. And when we probed Wnt signaling with small molecules, we can attenuate some of the differentiation. However, you'd never lose all of it. But when we took this secreted frizzle-related protein, which is something that modulates the wingless type signaling in the extracellular space, and so this is a soluble protein that will just bind to Wnt's outside of it, we see complete loss of the SOC17 endoderm specification when treated with SFRP, suggesting that this is a key regulator of this process and maintenance of SOX2. And so the picture that's been emerging from this is that if you confine these pluripotent stem cells, what you do is that interface imposes a certain stress on that population of cells, which leads to a multi-layering, which triggers some of these gastrulation-like differentiation events within that colony. And our gene expression analysis suggests that Wnt5A and Wnt11 are intimately involved. We see over 200-fold increase in Wnt5A. And if we knock that down with secreted frizzle-related protein, we can attenuate some of these differentiation outcomes. And so that's an evolving story that we think is quite exciting and will lead to platforms where you can build models of embryonic development in the laboratory to probe various questions of interest. And so, from this, we've been interested in, what are the next steps? And so, one of the first things I was curious with is, well, we can pre-pattern these. What happens when we take them off? And so we took these cells and we removed them from the substrate. They're not attached too strongly just because of the nature of the interface on these hydrogels. So you can just remove them quite simply, and then you can put them in a non-adherent dish and watch them grow. And the thing that's quite striking is that when you take cells that have been cultured on glass or plastic, they form embryoid bodies, as you would expect, very uniform spherical aggregates of pluripotent stem cells. While when you take the cells that have been primed on these micro-patterned hydrogels, we see a really interesting evolution in structure, where we get these multi-lobed aggregates of cells. And if we then do some immunofluorescent staining of these interesting markers, what we see is these multi-lobed cells are expressing differential markers associated with gastrulation-like events, suggesting that this pre-patterning priming is triggering some differentiation in these cells. And we're a biomaterials group, and so we get philosophical about the materials in a developing embryo and how that influences these processes. And so we took a number of materials and we encapsulated these embryoid bodies, and some of them seem to restrict this differentiation, like this example here of hyaluronic acid methacryl oil, which grabs these things. And then in the case of alginate, which allows cells to move around and proliferate due to their relatively weak ionically stable bonds, we see maintenance. And so this is something that I think is really exciting. And I draw parallels to early development, where the embryo implanted in the uterine lining experiences these biomechanical cues. And if we can trigger these events and control them and guide them in the laboratory, then we can build better tissue models using these types of techniques. Okay, so at the heart of both the mesenchymal stem cell story and the iPS cell story is this idea of, can we utilize materials chemistry approaches that are better tools to guide cells to have a very precise type of heterogeneity, and can this then be useful for biofabrication? And so that will lead to the next area that I wanted to present today, which is how we've been trying to tie this idea of stem cell engineering into assembly of tissues utilizing biofabrication approaches. And, I spent a lot of years, not wanting to get involved in 3D printing because, there's a lot of really just fantastic work, both at Ali Kadam Hosseini's lab, in Jennifer Lewis's lab, and many, many others. I could spend the rest of my time just mentioning all the leaders in this field. But what really triggered my interest in this was work that came out of Adam Feinberg's lab at Carnegie Mellon, where he developed this technique, free-form reversible embedding of suspended hydrogels, so-called fresh bioprinting. I'm assuming many of you have heard of this. And, back in, I think it was 2018, Tom Molly, a really terrific graduate student in the group, expressed some interest to go to this bioprinting meeting, along with Stephanie Nemec, another really great graduate student in the group. And so I sent both Stephanie and Tom to Carnegie Mellon to this DIY workshop that Adam puts on every few years. I think he's still doing it, so you should check that out. And so what is involved in this technique is essentially, you have this suspension of microgels. And so these are microscale gels. They can, trying to make sure I can click this video again. Yeah, no, it's not gonna let me. These are microgels that, essentially form a jammed suspension that this jammed suspension you can use to print materials and cells within that suspension. And Adam's done some really beautiful work to show that you can get very high fidelity structures in these microgel suspensions. And so even, I think it was last year, he showed in ACS Biomaterials Science and Engineering, you could print an entire human heart out of collagen with anatomical microstructure. And but one of the things that we were really fascinated with is that rather than dissolving that gelatin bath after you do your print, I started thinking, well, this extracellular matrix surrounding the cells could be utilized for as a tissue scaffolding. And so this is really gelatin, which is denatured collagen. Can we make this do things for us? And so the first thing that we wanted to explore is, well, can we put cells in this bath? And remember, if you heat it to 37, this thing's going to dissolve and melt away. And so one of the things that we were curious about is, well, can we stabilize this gelatin microgel bath and thereby allow it to stick around and culture cells on it? And if we do this, and we have various chemistries to stabilize the gelatin microgels, but cells just love it, right? And so they'll sit and adhere to gelatin. There's adhesion binding sequences in this material. And so we've been exploring ways that we can utilize printing in the gelatin baths in the conventional way of fresh bioprinting, but also stabilizing and using things like gelatin methacryl oil, which I know that that many, and Ali did really pioneering work in this area. And so this is really the approach. And so, initially we just polymerized the microgels, but then they remain a little bit diffuse and they'll scatter when you are trying to do things with it. And so Tom did some really nice work to show that if you add a liquid filler, and so essentially a 1%, very low fraction of gelatin methacrylate to the microgel suspension, you could essentially stitch them together I think of it like, spaghetti and a sea of meatballs, I guess. If you then go on and characterize the rheology, and so here you see that with the UV treatment, you get a large increase in the storage and loss modulus. And if we do this as a function of strain, you get these classical characteristics with crossover, showing that this is a elastic material. And importantly, these microgel suspensions made of gelatin methacrylate maintain their shear stress, their yield stress properties. And so if you look at the complex viscosity as a function of shear rate, you'll see that you have this nice relationship that relates to the definition of these non-Newtonian fluid baths. Okay, so why do we care? Well, we can do interesting things with them. And so Tom went and put cells in these gelatin methacryl microgel suspensions, as well as in a pure GelMA bath. And you can see that cells get very different morphologies over time. And so over about a week of culture, when the cells are cultured in these GelMA microgel suspensions, they elongate, they get very large changes in area. They really enjoy, sitting on these surfaces, and they migrate around and move. And we did some morphometric analysis and show that there's a considerable difference in the cell's characteristics as they're grown on these things. But at the end of the day, we were interested in, well, can we do more sophisticated things? And so we started changing the weight fraction of GelMA that we were putting in these suspensions towards making gradients. And so we found that we could change the modulus from just a couple kilopascals up to hundreds of kilopascals just by changing weight fraction. And using very simple techniques like having a divider between these two proportions, we could mix them together, as shown by these multicolored gradient baths. And so this is sort of the idea where, if you have this bath, you can 3D print structures in it. You can also have cells in the surrounding milieu. And by changing the characteristics, you can have a whole suite of different types of microgels within these suspensions. And really my vision with this is that if you can precisely tune the chemistry of microgel populations and then mix them together in well-defined ways, then these can serve as small microscale encoded extracellular matrices that can trigger various activities in cells. And so, we've been exploring that. Here's a nice picture of going from these large microgels to these small ones in a gradient bath. And as I showed, there's very simple ways to do this. Of course, you can have more complex methodologies for mixing, but I love simplicity. It's always a good place to start, I find. And so we started thinking about this in the context of our mesenchymal stem cell differentiation, and as well as our iPSC differentiation. And many years ago, I had worked with mesenchymal stem cells and understanding how the properties of their microenvironments trigger differentiation, and motivated by some classic work by Chris Chen, as well as some of the work that I did, looking at micropatterning cells, where we essentially just add one bottle of fat promoting cues and one bottle of bone promoting cues, and then let the cells decide which outcome they're going to prefer given their microenvironment. And this technique's been adapted by many over the years. This is classic work by Murat, and when he was with Jason Burdick, showing how you could tune the microenvironment and let the cells make the decision when giving a mixture of soluble cues. And so we use the same approach with the microgels. And the first thing we asked is, well, if you change microgel size, will you influence the way they differentiate? And so this is using an adipogenic media, an osteogenic media, and then a mixed media where you have both cues, as well as a control. And what we find is that when you culture mesenchymal stromal cells in these microgels over the period of several weeks, that they tend to differentiate, and that the propensity to differentiate to one lineage versus the other depends on both the media supplements as well as the microgel environment. And if we quantitate this FABP4, fatty acid binding protein 14 marker, as well as oil red O as a histology stain, we can discern the degree of differentiation as a function of microgel size. And so in this case, it suggests that the small microgels are better for adipogenic differentiation than the medium ones or the large. We also did this for bone differentiation. And what we see is that when you culture them for several weeks, again, we can see subtle differences. You can see the alkaline phosphatase staining here. These these black stains in here is actually mineral. And so the cells are depositing mineral over the period of, I think the final time point in this experiment was 21 days. And, and so from this experiment, we found that the medium and the large microgels seem to be good for bone formation. And so this gives us a way to tweak our microgel properties to derive various lineage outcomes that might be useful for making things like multiphasic grafts, where you want to 3D print something where you're going to have bone and cartilage and fat and tendon and other tissues spatially with temporal control in these materials. And so here's just a video showing some of the matrix that's accumulated. And so this is a CT image done by TT Hung at the Bioresource Imaging Laboratory at UNSW. And so this microCT clearly shows that we have calcium deposits within the microgels. And so we were actually quite surprised. I think my thought was that it would form in these pore volumes, but I guess, it does make sense. If you've got this gelatin collagenaceous template within those gels, this is going to be a preferential spot for hydroxyapatite and other minerals to accumulate. So that's all well and good. We can tweak these things, but another thing that we've been excited about is it possible to make vascular structures within these materials? Because whether we're making bone, cartilage, any of the tissues that we might be interested in, we're going to need blood supplies. And so we're interested in if we could do this in these baths. And so we borrowed a page out of Jennifer Lewis's playbook. And so some years ago, she did this very elegant work printing this thermoresponsive pluronics material within hydrogels and showed that you can make complex vascular structures. And so we asked if we could do this in our baths. And so here's some rheology of pluronics. And so it's thermoresponsive. And so you can print it at one temperature where it's a solid, and then change the temperature and it then becomes a liquid. And so we were curious if we can make channels this way. And so here's the workflow. We have our gelatin methacrylate particles. We print the pluronics into the microgel suspension. We can then photopolymerize the GelMA particles surrounding that pluronics and then suck out the pluronics and thereby leaving a hollow channel. And so we're able to do this, and you can see here, we don't get the most beautiful, tight, well-defined lines. This is just a property of the mismatch between the microgels and the pluronics. But hey, we can at least get structures that you can flow fluid through and perfuse, as shown here by some of our ink structures. We can also characterize these quite robustly. And so once we print these channels, we're able to do microCT imaging. And so because of the contrast difference between air in the channel and the surrounding gel, I thought this was a really unique way of characterizing these. And this is an example of some of the channels that we've printed with our pluronics ink. And because we can get these well-defined structures, we can do modeling. And so Tom did some really nice flow modeling. And why I show this is that I think, even if you don't make a perfect, well-defined channel, I mean, nature doesn't really make perfectly well-defined channels in our vasculature either. And if we understand the kinks and the various flow regimes within those structures, and we understand how fluid will flow through there, then we can relate that to endothelial cells lining that, as well as exterior processes like angiogenesis out from these regions of laminar flow or turbulence, what have you. And so I think it's a very useful platform. And we published this just a few months ago in Biomaterial Science. So have a look if you're interested. So as I said, now that we can characterize it, we can start studying more complex biological processes. This is ongoing. We've done some endothelial lining, and we can form some pretty nice vessels. They're still somewhat leaky. I think this is a problem spanning all of the field, but we can get barrier functions to a degree where we can study some biology now. And so we've been doing these for various reasons. Here's an example of a melanoma model for some of our cancer research, where we've made this bifurcated channel, and we've got HUVEC endothelial cells lining the channel wall. And then we have these melanoma 3C6 tumor cells that we've printed in there. And then in the surrounding bath are adipose-derived stem cells to serve as a classic fibroblastic stromal cell. And so in this way, we can bring multiple cells together to make these complex microenvironments. And so I think there's a lot of exciting things we could do. And, it's It's not the most simple platform. It's also not the most complex platform. So, it's really been our mantra over the last several years, is trying to meet somewhere in the middle so we can have enough control to study these questions, but while having enough complexity to recapitulate complex biology. Okay, I wanted to finish with something that is kind of an emerging area in the group, which is following on from this fresh printing, which is can we print more than just protein and polymer-based inks into these baths? And this came about by some really nice collaborative work with Iman Rouhani, a researcher in chemistry at UNSW Sydney, who he's a bone tissue engineer and had done some very nice work developing ceramic materials for bone. And he had developed a new paste, a calcium phosphate-based material that could rapidly solidify in aqueous environments. And we thought this was quite exciting because, if those of you who are familiar with bone tissue engineering, the classic approaches involve very high temperature sintering of green ceramic materials. And so if you 3D print a material, you've got to raise it to thousands of degrees, often using toxic additives. And so this is not really beneficial to biology, of course. And so, this ink that Iman had developed, we thought was quite interesting. And we were wondering, can we actually print this in our material? And so here's some characteristics of the ink. As I mentioned, it's a calcium phosphate nanoparticle paste that has various chemical additives that essentially give it this property where it can nanocrystallize within minutes once it's exposed to an aqueous environment. So it's in a glycerol solution. Once it's put into water, this water at the interface catalyzes the formation of the hydroxyapatite phase, and you get these nanocrystalline hydroxyapatite needles that come out from the material. And it's quite fascinating because within 5 minutes, you're getting properties similar to cortical bone within a petri dish. And so Iman saw Tom and Stephanie in the lab 3D printing in our suspension of gelatin. He started wondering, well, could we do this with this ceramic ink? And so this led to a new project. We're exploring this. Before I tell you about that, I do want to mention some of the advantages. And so with this ink material, because you don't need to do sintering, you can do it in the presence of biology. And so you can encapsulate proteins within the ceramic structure, and they essentially get interlocked with these nanostructured hydroxyapatite needles. And over time, it can release. And so you can encapsulate antimicrobials, bioactive materials. We demonstrated this with ibuprofen and dexamethasone release from our ink. And these properties of being able to release drugs as well as maintain mechanical integrity really gives a lot of advantages to the material. And so, we were curious if we could freeform print in here. And so Iman went and worked with Tom and Stephanie and tried this in our gelatin microgels, and it works really quite well. If your yield stress bath properties are such to stabilize the hard material that you're printing, then you can make all sorts of complex architectures. And this is really just someone flicking. I think it's Sarah Romanazzo, who is a postdoc, the lead author on the subsequent work I'm going to show you. And you can see that there's a very tight interface between the nanostructured ceramic and the gelatin. We then explored the types of structures you could print, and it's really all sorts. And so using hand printing through a syringe, as well as using a custom extruder on a robot, Iman and Sarah were able to print these osseous labyrinths, trabecular bone-like structures, helical haversian canals, things that I'm told are really complex bone structures that are important for tissue engineering. It's not my area of expertise. And using the traditional fresh approach, you can harvest these and pull them out of the bath, as I've shown here. And so here you can dissolve the gelatin and pull out your ceramic material. But we're also curious if you could do it in the presence of cells. And so we did this with, in this case now, the mesenchymal stromal cells from adipose tissue. And we've coined this term ceramic omnidirectional bioprinting in cell-laden suspensions, COBICS. And with this COBICS technique, we can print in the presence of cells. There's high cell viability, very little cell death. The cells grow. You can see in this day 14 image, this black space is where the ink is printed. These are the mesenchymal stromal cells fully adherent to proliferating in the microgel surrounding that. if we do characterization of the cells within the material, we find that proximity to the calcium phosphate ink is enough to stimulate osteogenesis. And so I suppose not too surprising for those working with stem cells in bone microenvironments that when they see that calcium phosphate environment, it triggers signaling that stimulates osteogenesis. And so we see a lot of osteoprogenitors at early points, as indicated by RNA analysis. We did some transcript profiling of RUNX2, osteocalcin, osteopontin, et cetera. When you add osteogenic media to further stimulate that, you get much higher degree of differentiation. In fact, they start mineralizing the entire scaffolding. And so we think this is an exciting way to make bone-inspired microenvironments for disease modeling, as well as the thing that we're really excited about now, and we're looking at potentially commercializing this, is being able to print these materials in an operating room in a patient. And so without having to sinter, you could imagine that's future vision where a patient gets a CT scan, MRI, and you image their bone. You can then translate that architecture into a 3D printer's STL file. And then after resection of diseased bone or after an injury, you could then directly print within a patient these bone structures, utilizing perhaps their own autologous stem cells. And I think that's a very exciting future. So, of course, we wanted to check if these materials are biocompatible in vivo. And so we've recently done a few studies in a critical size defect in rabbits. And so we compared against a very standard calcium phosphate material from Medtronic that's utilized widely and is FDA approved, these master graft granules. After 3 weeks, we see no signs of inflammation with the implant. And in this case now, we haven't 3D printed. We just put the ink directly into the defect site. If we then do some micro-CT scans, you can see this is the predicate up here in the upper left, and these are granules, so it's got quite a different image on CT. But we see no overt signs of inflammation. And if we zoom in, intriguingly, at the 3-week time point, and we just sacrificed the 6-week time point, and so we should have some more preliminary data shortly, but we see some evidence perhaps of cortical bone growth at that interface, suggesting that not only is the body tolerating our material, but it's actually stimulating further bone growth at this interface. So we think that's quite exciting. All right, so that's what I wanted to show today. In summary, I hope you appreciate how the importance of materials in directing cell activities. I think that there's a bright future in integrating and fusing these disciplines together. Using various fabrication approaches, we can print materials in the presence of cells. We can involve multiple cell types. And so I think we're getting much closer to a very exciting time where the cell engineering, the understanding of the mechanobiology and the epigenetics can be aligned with our advanced materials fabrication approaches to make better tissue models. And with that, I'd like to thank the group. I've mentioned everybody throughout. And so here's a nice picture of us that we took. I've got to thank the funding, of course, from the Australian government as well as the NIH. And with that, I'm happy to answer any questions. Thank you very much.
Thank you for the great talk, Chris. I appreciate it. Several questions came in. I'm just going to read them. Very interesting work. How is the resolution of your GelMA-based bioprinting and ceramic-based bioprinting?
Yeah, the resolution currently is not the best. And so, we get down to the tens to hundreds of microns range. The key limiting thing currently is that our microgels are pretty polydisperse. We use a very simple approach with an oil and water emulsion type process to make these microgels. And as I said, I love simplicity. And so it works enough for us to put these things together and then study how they interact. The future, of course, is going to be increasing that resolution. And Adam Feinberg, he has a company, Fluid Form, that's done a really beautiful job in designing new approaches for developing microgels. And you look at Adam's work, and he has new ways in using microfluidics and advanced detergents. You can get very monodisperse particles, and in doing so, you can get much higher resolution. And I can't think off the top of my head how low they go, but we can certainly improve. For our work, we haven't that hasn't been a primary focus. We've more been interested in, can we make new chemistries in these gels? And so we're not only looking at gelatin, I just showed you that today, but various types of active biomolecules, making microgels with them. And I think this is a lot of the future of this work. Jennifer Lewis, for instance, published last year a really interesting paper where she used organoids as the printing material. And so I think it's an exciting future.
Thank you. Mostly 2D based studies have been done, but you show 3D bioprinting based platform. What similarities and differences have you seen using both 2D and 3D platforms?
Yeah, the 2D versus 3D. This is the thing that wakes many of us up in the middle of the night. Yeah, we see lots of differences, to be frank. And this has been well understood now for a time, and I didn't harp on this. I do get on my 2D versus 3D soapbox. The reality is 2D is here to stay. It's a very useful way to look at cells and to ask questions about them. However, I absolutely acknowledge that we live in a 3D world, and cells signal very differently in 3D microenvironments. And so really our main intention is to bridge this divide. And so 2D plastic, I mean, cells do some very strange things. 3D tissue, they behave in ways that are more clinically relevant to us. And so if we can get there in the middle and make these 2.5D pseudo 3D architectures that we could probe with the tools at our disposal, 3D bioprinting, advanced imaging, and use that to guide our understanding of 3D. That's really where I reside with this. To be specific, for instance, the mesenchymal stem cells, they adhere very differently. Some very beautiful work from Nate Hupsch and David Mooney showed some years ago that they adhere through different integrins in 3D environments compared to 2D. They perceive the mechanics quite differently. And so you have to be very careful when you make conclusions from 2D. But my mantra is that if we control our microenvironment appropriately, then we can make connections between the observed activity and things like mechanics and chemistry to inform how we interpret the biology. But it's a great point and something that we could talk hours about. We see differences all the time, and we need to keep in mind that there will be in every biological system we explore.
Thank you. For the reprogramming process, the colony formation seems one of the important features. Do colonies form on the patterned surface?
Yeah, no, it's, in fact, the way I'm thinking about, these iPSC colonies, quote unquote, now, is that, they tend to have a natural tendency to aggregate. And this is a property of the cells, right? They really like to express E-cadherin and form these sheets on the surface. And so these are human pluripotent stem cells. So they're the primed pluripotent stem cells, if you will, rather than the naive. So one of the things that I'm really intrigued about is that when we confine them using micropatterning, we essentially force them into an aggregate structure. And because they keep proliferating, they start to move into 3D. And this is speculation, but we're currently exploring it. I feel that this confinement essentially tells the cells that they're in an environment more akin to their native embryonic architecture. And this then catalyzes signaling events that are related to embryogenesis. And so, the vast majority of work that's been done has been in monolayers with human pluripotent stem cells. And I think that we've developed conditions to ask the right questions and to get some really nice biology done in those formats. But now moving into these more sophisticated platforms, we can ask more sophisticated questions and promote more sophisticated activities. And in our case, microconfinement, I believe, is triggering this multi-layering that we see at about 48 hours and the appearance of the primitive streak. And so I think it's just absolutely stunning that utilizing microengineering techniques that were initially adapted for making computer chips and deformable hydrogels, bringing these together can trigger events that underscore early embryogenesis. So it's, I think, pretty exciting. I hope that answered your question.
Yeah, it did. Thank you. Compared to soluble factors, do you think biomaterial-based factors play a bigger role in regulating cellular behavior?
Yeah, no, it's, I'm one that believes that they're all so interconnected. I think that soluble factors probably play a bigger role if you're keeping everything equal. If you're putting the cells on a glass surface, for instance, we certainly see this. The thing that I love about tweaking the biophysical factors is that what it does is it puts cells in a context to receive those soluble factors differently. And this is a paradigm that I'm really excited about. And so the idea that in a microenvironment, let's say you have a stiff matrix and the cells see some growth factor. And because the pathways linking feeling of stiffness and integrating that morphogen are often intertwined. So a lot of the downstream effectors like mitogen activated protein kinases are triggered by both mechanotransduction and soluble factors. And so if you change that microenvironment, you change the way that the cells can interpret those soluble signals. And so it's that interplay, I think, that is absolutely critical. Whether one is better than the other, I mean, it's very tough to say. I think in different contexts, physical properties play a bigger role, but in many, many examples, I think we're all still very soluble signal centric. That's a hard one to say because signaling gradients using morphogens and cytokines is so critical for so many biological activities. But it will not, and this is so important, it will not trigger the appropriate form and function without the material's properties. And so those have to be very specifically linked. Thanks for that question.
Yeah, thank you. Is it possible to regenerate both osteal and chondrial tissues in a bioprinted scaffold simultaneously?
Yeah, no, that's an awesome question. I wish I had data to show you. We're looking at this right now, and I encourage others to do so as well. I mean, there have been, people have shown, for instance, Hala Zreikat at Sydney did some very nice work to show that when you have a very hard ceramic material and then like a fibrous hydrogel-based material in conjunction, and you put a stem cell population on there, the cells will naturally start to turn bone on the calcified ceramic material, and then form tenocytes on the fibrous material. And so I think cells have the propensity to make these multiphasic structures given the right types of cues. In our lab, we've been using this Cobix where we print within the microgels, and then we've been adding a few soluble factors. And so that these progenitors, if they see the ceramic ink, they'll be predisposed to go to bone. Well, if they see the microgels, they may want to turn into cartilage. And so I hope we'll have that data soon. We don't yet, but that's the hypothesis. And we hope that by integrating these tuned microgels with these 3D printed ceramics, and it's all in the interface, and with defined medias, I believe we'll be able to form all sorts of gradients of tissues. And once we can get vasculature penetrating that, I think we have the potential to make true useful grafts that have ceramic materials, hydrogels, multiple lineages. It's an exciting time.
Thank you for your extraordinary presentation. Why was the ceramic material beating? Is that just a bone or something else in any other cell culture?
Yeah, I went rather quickly through that, and I apologize. Now, the beating was because we were actually flicking the scaffold to show how tight that interface was. I'm sorry for the confusion there. It wasn't beating. I am interested in cardiomyocyte biology, and so we may put some beating cardiac organoids in there at some stage. But in this case, we were just moving it to see that there was a very tight connection, we believe, because the hydroxyapatite nanocrystals were essentially interpenetrating with that gelatin interface to lock in place, because it gets actually pretty tightly locked.
Thank you. Is there a need to match the viscoelasticity of the ink with patient-specific microenvironment viscoelasticity for a better biomimicry?
That's a great suggestion. I mean, I think that's the direction that we all hope to go, where we can control as many patient-specific parameters as possible. If the tools are there, and many of them are, where you can discern the architecture of the patient's site, the mechanics of the patient's site, the cellular identity of the patient's site, all of the immune. We didn't even talk about immune cells today, but the immunoregulatory aspects of that. These are all things we should pay attention to. I could imagine that if you remove a defect from a patient and do an appropriate mechanical test, you can even query the chemical composition of these materials and then tweak your formulation appropriately. I think it's absolutely possible to do patient-specific graft design in that way. Currently, I think the limitations are that these techniques take time, and that could prevent rapid adaptation of that, but I think it's certainly possible.
Thank you. Does it have to have a methacrylate group in order to be able to cross-link with UV? Isn't there an alternative way that doesn't require chemical changes in the material?
Yeah, absolutely. We've had a lot of luck with GelMA. I know many in this audience work with GelMA. Thanks, Ali. But we've used other methods. We use the first one we did for the Cobics, and in that advanced functional materials paper, we just used glutaraldehyde to cross-link. Again, this is a non-natural cross-linking method, but certainly there's other approaches that you could use. Enzymatic, you could imagine using lysyl oxidase, some of these enzymes that are used in biology to cross-link collagen-like architectures. So I think going on to enzymes are better, and it's a good point because one of the things that we've been recently focusing on is the degradability. And with these stabilized microgels, when you're using GelMA or something like glutaraldehyde cross-linking, they're almost too stable. And so because of that unnatural cross-linking, it's very difficult for matrix metalloproteases to digest the material. If you decrease the weight fraction such that you have more room and more porosity and the degree of functionalization, and so we've actually tried to do poorer functionalization procedures so that we have less cross-link points, but then you have a softer material. So there's different places where you have to sacrifice, but there's certainly all sorts of means that you can modify these outside of using GelMA.
Thank you. What is the effect of biomaterial length scale? Difference between hydrogel scale, low nanoscale versus nanofibrous protein scale, high nano versus microscale fibrous material form.
Yeah, those hierarchical relationships are stunning. I mean, it's one of the things that got me really excited about biological systems. And so, I mean, it's all interlinked. The nanostructure influences the way cells can form focal contacts or focal adhesions in 2D. This then links to how the cells signal to their neighbors to form more macroscopic cellular assemblies. These tissue assemblies are often residing on things like membranes, like a basement membrane, or larger scale structures like bone. And so, as I think of it, I think it's a cascading effect that's intricately linked from the molecular scale, and so it even goes sub-nano, where precise moieties in the extracellular matrix that correspond and are recognized by precise receptors at the cell's interface. The way that those interactions augment activities ultimately reside in how these cells assemble to form tissue, how these microtissues then assemble to form whole organs. It's all interconnected, and it's such a complex, beautiful picture, and so complex that I'll never get bored. There's, as a field, I think we always have an amazing set of challenges to explore.
Thank you. Have you seen mechanical parameters influence cell migratory behavior?
Absolutely, yeah. Durotaxis, all of these events. It's something we've been exploring in these gradient baths, and certainly, it hasn't been as pronounced as we'd hoped. We're still tweaking it, but I think it's a good platform if you're interested in studying these types of things. And so we have seen cells migrate towards stiffer materials. Certainly we've seen this in our 2D gel systems, particularly mesenchymal cells, where they'll have a tendency to want to migrate on a stiffness gradient. In the microgels, we hope to be able to see this. We haven't explored it too much, but I think one of the things that I didn't present today that we're quite excited about is our cancer models, and where if you can have a gradient of stiffness in microgels, gradient in size, chemistry, maybe having a hot spot secreting some chemokines, then you could really try to decouple these complementary effects that influence a cell's propensity to migrate. And migration in the cancer context, of course, is looking at things like metastasis, and we're exploring that. But for stem cells and for other types of somatic cells, it's equally important for processes like epithelial to mesenchymal transition during morphogenesis. So we see it, and many others have as well.
Final question. What sterilization method are you using for your implants or 3D printed materials?
Yeah, well, for the implants, for the Cobics material, we did gamma. And so gamma is a pretty standard approach. And we believe that gamma would be better to maintain the integrity of the material rather than using something like the ethylene oxide treatments. And so gamma sterilization for the rabbit studies, we may explore others in the future, but that's the one. For most of our in vitro stuff, we just use sterile reagents when we prep the materials and usually leave it under UV light briefly so we don't modify the proteins considerably.
Excellent. Well, thank you for your time and your great talk, Chris. Appreciate it.
No problem. Thank you so much for having me.
Have a nice day.
Yep. Bye, everyone.
Bye-bye.